saw that you had another meetup with a similar theme last week or so with Mike Love.
Yeah, 2 weeks ago.
2 weeks ago.
Yeah.
And then Stephanie is going to present.
Yeah. We are very happy, actually. We have very good invited speakers. You accepted our invitation, and it's a great pleasure for us.
Yeah, in many ways, it's great that the pandemic has opened these opportunities, right?
Yeah, actually, that's what we wanted to open, because we don't have these opportunities so in Tunisia. And so that's why we wanted not only Tunisia, but African, and from all over the world. So
yeah, absolutely.
Okay, so I think in 1 minute we'll start. So really, you're also an inspiring speaker for us, because I saw that you did your master in botany. So your background, is like biology, because so somehow, well, not, but.
Actually, my worst courses in college were in statistics and computer science. So I think I just keep doing the same things that I didn't do well at before. I keep trying. I'm sure I can do better.
Yeah, everyone, actually. We are all learning.
Exactly.
Okay, so hi, everyone. Yeah, that's good that you put your background also. So we encourage all the biologists and bioinformatician and computer scientists to put their background, so we can see the background of everyone. So hi, everyone. So welcome to our study R for Bioinfo. This is our second workshop. So it's entitled Introduction to Annotation Resources in Bioconductor. We have the pleasure to have with us Professor Martin Morgan. Thank you, Professor Martin Morgan, to have accepted our invitation and to be with us today. And so I will introduce Professor Martin Morgan. Professor Martin Morgan earned his undergraduate and master degree in botany at the University of Toronto. So I'm glad that, it's like to show to all the biologists that you can be like good bioinformaticians. Dr. Morgan did his PhD studies at the University of Chicago, and then so he spent 10 years as an assistant and then associate professor at Washington State University. So in 2005, Professor Morgan worked on the Bioconductor project for the analysis and comprehension of high-throughput genomic data. He has led Bioconductors since 2008. Professor Morgan moved to Roswell Park Comprehensive Cancer Center in Buffalo, New York in 2015, where the Bioconductor project is now based. Thank you, Professor Morgan, to be with us today and to introduce us to the annotation resources in the Bioconductor project. But before moving on, so we want to introduce you also to the R Ladies initiative. So I will start sharing my screen. Let me see how to share my screen, actually. Where is it? Okay. So we have.
Hello and welcome.
Before, sorry. I don't know how to share my screen here.
It's at the bottom. at the left, right. Sorry.
Okay.
You see?
Yeah, good. Thank you.
It's,
So can you see my screen?
yes, that's okay.
To this workshop session. Hello and welcome to this workshop session. Before starting the workshop, we'll introduce you to the Our Ladies Global and Our Ladies Tunis initiative. The R community suffers from an underrepresentation of minority genders in every role and area of participation, whether as leaders, package developers, conference speakers, conference participants, educators, or users. As a diversity initiative, the mission of Our Ladies is to achieve proportionate representation by encouraging, inspiring, and empowering people of genders currently underrepresented in the R community and to facilitate individual and collective progress worldwide. Gabriela de Caros founded Our Ladies on October 1, 2012. Our Ladies Global was born and the grant was awarded in September 2016. Since then Our Ladies has grown to 170 chapters in 44 countries and 39,000 members. Our Ladies Tunis is part of Our Ladies initiative and was created in May 2020 by women working as data scientists and biostatisticians and bioinformaticians. Our goal is to create an R community in Tunis and empower underrepresented genders in the R community. This is the core team of Our Ladies Tunis. Come and join our community on social media. This presentation was made with R Markdown. Thank you for watching this video and enjoy our workshop.
Thank you for your attention.
Yeah. So I will be moderating this session. So I'm Dr. Hedia Tnani, and I work as a bioinformatician at Pasteur Institute of Tunis. And now I will step back to let Professor Martin Morgan introduce you to the annotation resources in Bioconductor. Thank you.
Thank you for the introduction and for the really great work that I think Our Ladies is doing. It's really wonderful to see a diverse group of people on the call and to share some of my enthusiasm for R and Bioconductor. I've seen a few comments about your background and so on. I don't really know who you are in person. And of course, it's really different, doing a Zoom webinar instead of an in-person one. But I'll try and not go too quickly. And I hope that you can add things to the chat or ask questions or, unmute yourself and talk, whatever you feel comfortable with as we go along. Maybe to get going, I'll just paste into the chat the document that I'm going to be using. I should have prepared this for you earlier and given you a chance to read it and get up to speed, but actually it didn't exist earlier. But it does now, so that's great. Yeah, so I guess I'll switch over to presenting, and I'll try and keep track of the chat. And as I say, don't hesitate to interrupt. So I'll just switch over to my RStudio session, and I hope that you can see that.
Yes.
It's good.
Yeah.
So I've got a link here in case you missed it, mtmorgan.github.io/annotation resources. And what you've got at that location is what I've got over here in this window. And this is kind of like my little plan for today. We'll see how things go. I'm still just kind of adjusting things here, figuring out how to get things just so. Okay, let's try that for a while. Yeah, so I'll say a few words. I think I can make this a little bit bigger. I'll say a few words of about where we're going. I'm going to try to make as few biological or computational assumptions as possible. I know that Mike Love spoke at a recent meetup and covered DE-seq2 for bulk Rna-Seq differential expression analysis. I'm sure that some of you were there and that some of you weren't, but we're kind of going to build on that a little bit. My overall plan is to provide a quick review of bulk RNA sequencing, primarily to introduce this airway package and sample data set. These are links to what I call Bioconductor landing pages that describe the package. And then we'll talk about types of genomic annotation, identifier mapping, gene annotation, working with gene sets. And if we had more time, I'd really like to get into cell annotation, which is really important in the single cell RNA sequencing technologies that are coming up. But I don't think I actually have time for that, so we'll stick with these topics here. I'll say just a few words about Bioconductor. It's a kind of a collection of 1900 R packages that have as a kind of common theme the idea that they're to help with the statistical analysis and comprehension of high throughput genomic data. It was started in 2002, I think, so it's been around for quite a while. It's pretty heavily used. There are actually more than 500,000 downloads to unique IP addresses every year, and there are like 40,000 PubMed Central citations. And the development of R and of Bioconductor is supported by the US government, the US National Institutes of Health, National Human Genome Research Institute, and the National Cancer Institute. So it's all of this good work actually doesn't come out of thin air. It's actually a paid development on the part of the core team, but then also a huge contribution from our community of users who end up writing R packages that are then used in development. And they contribute to Bioconductor. So this is a link to the Bioconductor website, and then we're similar to CRAN, the place where you install R packages, but just a little bit different. And we have a package called Bioc Manager that we use to make it easy to install Bioconductor packages and actually CRAN packages. So if you're following along, you could try and copy and paste this text into your R session, and for you, probably it'll actually install the Bioc Manager package from CRAN. And then we're going to use a couple of packages, dplyr and airway. And if you wanted to follow along again, we'll use a couple of other packages as well. You could take those packages, that vector of packages, and then install them using Bioc Manager. And I'll just point out a couple of things, that this airway package is a Bioconductor package, but dplyr is a standard R package. And you can install either type of package through Bioc Manager. So we've got a little bit of a quick introduction to Bioconductor, and then this special instructions for how Bioconductor packages are supposed to be installed. Is that good? Are there any questions? Do people want to chime in at all?
Maybe we have to send them, to install, and then, wait a little bit. Can we put in the chat? Yeah. Okay. Okay.
if you've got to this place here where you're installing packages with Bioc Manager installed, that's probably good. It'll probably proceed, take a little while, but hopefully there won't be any real challenges with installing those packages. So I think what I'll do is I'll just
Okay, so I put in the chat.
perfect, yeah, great. So what I wanted to do was in this chapter, I just wanted to provide us with a little bit of biological background and the way that this type of data is represented in R. And again, if you're at Mike Love's meetup a week or so ago, this will be familiar. So the idea is that what we want to do is measure levels of gene expression, maybe in different tissues of an individual's body, like cancer and normal tissues, or maybe in two experimental groups of mice or something like that. And we want to measure gene expression for many thousands of genes. And the technology that does this is called Rna-Seq. And the way it works is genes, which are encoded in RNA, encoded in DNA, get transcribed and transcribed into RNA, and then translated into amino acids. And amino acids are the proteins that participate in cellular interactions. So DNA to RNA to amino acid to proteins. So at the RNA stage, it's possible to take a strand of RNA and actually sequence it. Sequence that RNA to sequence the RNA, and then to quantify how many molecules of RNA there were. And what you actually end up with is a matrix of genes and samples, and then each entry in the matrix is the number of, or proportional to the number of RNA molecules that were found in that sample for that gene. It's really pretty amazing. I see that Ibor says they have a message. Bioconductor version requires R version. Bioconductor version 3.8 requires R version 3.5. And I was wondering, Ibor, what version of R that you're actually using. It seems like it might be kind of old. You can do like, in R, you can say like R version string. I'll tell you how to do that. Yeah, this is one of the things that Bioc Manager is much more picky about, which version of R you're using with our version of packages, 362. Yeah, so I think that actually what's happened, Debor, is that you've installed new packages over your old package. And in the chat, I'll just say that what you should do is remove.packages Bioc Manager. And maybe also, just for good measure, remove.packages Bioc version. Then start again with install.packages Bioc Manager repo equals https and .rproject.org. And that'll likely get you to the next step without having to reinstall R. Your R is relatively new. And I'll also mention that that you might want to run Bioc Manager valid, which will tell you that maybe you have some packages that are out of sync with one another. And it'll give you some instructions for how to correct things. I'd leave that task until after the workshop, though. Maybe I'll just make a note here and try and remember to use Bioc Manager valid to check.
Yeah, you need to get true. So when you run the Bioc Manager valid, if everything is okay, you will get true.
True, right.
Yeah.
Otherwise, you'll get instructions like, I don't think my installation, I'm not a very good person here. I think it'll tell me that I have a whole bunch of packages that are out of date. Yeah, I've got 58 packages out of date. And it says, if I want to make them current, I can select all of this and paste it into the command line, and then it'll fix it all up for me, but that'll take a long time, and I'm not going to do that just at the moment. So I would encourage you not to do that either. All right. Okay, we better get moving. Although I think that's one of the great things about workshops, right? It isn't so much about the material that we cover in particular, but maybe learning about some of the problems that we encounter and watching other people do how they work can be helpful. So what I wanted to do was load this package called Airway. And, actually, an example of a type of package. It doesn't contain our software code. It actually is there just to provide data, so that the data can be used in workshops or for reproducible research. So the data that this package contains is accessible by saying data Airway. That loads the Airway data object into our R session, and then it actually creates an object called Airway, which I'll now display. So this is an essential object in the bioconductor world. It's called a summarized experiment, and it contains the results of one of these Rna-Seq experiments. And I'm going to access different parts of the data using these things called accessors. So there's an accessor called assay, Airway. And I'm going to ask about what kind of an object does this accessor return. It says it returns a matrix. The matrix has dimensions. The matrix is a two-dimensional object. The matrix has 64,000 rows and 8 columns. And that makes it clear that I don't want to do just this, because then I'd print out 64,000 rows, or a good number of them anyway, and 8 columns would just sort of spew all over my screen. So instead of what I'm going to do, I'm going to call head. And head returns just the first 6 rows of the matrix that is in my Airway object. So this is the first 6 rows of a matrix. And these are actually samples. There's a sample that's identified SRR103, blah, blah, blah. Another sample, another sample, another sample. And then these are genes. So this is a particular gene, another gene, another gene. And then these are measures of Rna-Seq, of gene expression in this sample and at this gene. So this sample had 679 units of expression. This sample had 448. This sample had 873. This sample had 408. So a measure of gene expression. And you can see already some interesting things. So here's this particular gene, 0005. It had no gene expression in the first 4 samples and no gene expression in the second 4 samples. So actually this gene wasn't expressed, or we didn't detect expression at all in this experiment. So this is the expression data. and it's the result of the Rna-Seq experiment. But actually, the Rna-Seq experiment has been conducted in a way like these samples have come from different experimental treatments. And the airway object contains information about the experimental treatments. And the information is available with another accessor called call data. And you'll notice there are 8 samples, and here we have 8 rows, one for each sample. The sample has an identifier that's the same as the matrix column header. And then there's information about the cell line that sample came from, and whether it was treated or not with this substance called dexamethasone. So it's some kind of biological experiment, and there's been an experimental design where there've been, you can look, 1, 2, 3, 4 different cell lines, and each cell line has been exposed or not exposed to dexamethasone. So that's like the experimental design. I'm gonna, just thinking about, how I work, and I wanted to share with you one thing that I find myself doing increasingly, and it's to kind of combine the approach like this, or the R command, where we did head of assay of airway. And I've started to combine that kind of approach with an approach that I found really useful for manipulating data. And it's the approach that's been made popular under the name of tidy data and the tidyverse, and in particular, this library dplyr. And so I'll just say library dplyr to load the dplyr library. And maybe just as I'm getting here, can I ask, you could maybe do, I don't think you can do thumbs up or anything like that, but maybe you can just say yes or no if you've come across the tidyverse before, if that's familiar to you.
In the chat, maybe, yeah.
Yeah.
We have one yes. Actually, don't be ashamed to say no.
Yeah.
We have 3 yes. Wow,
well,
many yes.
Yeah, cool. Maybe all the others are no's.
Yeah.
So far we have, excellent, yeah. Great.
Not yet. Good.
Yeah. Yeah, great. Well, it seems like some people have and some people haven't, and haven't used it much, but I think it's going to be totally great. It's really, really great. So here's my call data from the airway. And there are a couple of things that, it doesn't actually display that well. It sort of wraps around the display width, and if this were a big data frame with thousands of columns, it would have just spewed out all over the screen. It would have been completely illegible. And the way that the tidyverse kind of handles this is it has a kind of a data frame that's kind of enhanced, I guess. And they're called a tibble, T-I-B-B-L-E. And so I'm going to take my column data, and I'm just going to make it into a tibble, which doesn't change the data at all, but it just displays it in a different way as tibble. Let's look at the output and then go back a little bit at the input. You can see we've got exactly the same same columns and the same values, but we've only displayed a rectangular part. We haven't tried to wrap things around to get all of the columns in, and one of the columns didn't fit, so it's listed down here at the bottom as we've got another column. And then for each of these columns, we have a little annotation down here that says this column is a factor or this column is an integer. So we have a little bit more information about each of the columns being displayed. So the tibble is kind of nice, just like visually, it's much nicer to work with. Another kind of thing that I did, right, when I was working with the tidyverse was I used this thing, it's called a pipe. I think on my keyboard, it's not too bad to type a pipe. It's like a percent, greater than sign percent. I think on some people's keyboards, it's very difficult to type.
There is a shortcut, no, for that?
Is there? What's that?
There's a shortcut for the pipe.
How do you enter it, or?
I think, if I'm not wrong. Okay, one moment. So
Yeah, that's right. No,
let me see. I think it's Control-Alt-M, if I'm not wrong. Can you try it?
I have a Mac, so maybe.
Okay.
But maybe maybe Control-Alt.
I will open my R to check.
Okay. Yeah, it could be that there's a shortcut. That'd be pretty good. And actually, what this does is it takes. It's kind of just like some people call it syntactical sugar. It takes whatever argument is on the left-hand side, and it puts it in the first position on the right-hand side. So you could have written as tibble, call data, airway. That would have done the same thing. It's just that with the pipe, it seems like the way that you write the code is the same as the way that you're thinking about what you're doing. You take the column data out of the airway data structure, and then you transform it to a tibble. So you kind of write the code from left to right the way that you're thinking about it. Whereas with this structure, it's kind of, you work from the inside out. Control-Shift-M.
Yes, Control-Shift-M.
Excellent. Control-Shift. Yeah, there we go. Excellent.
I use it a lot, actually.
Yeah. Yeah, that's great. That's great. So actually, when we did this, we said head of assay of airway. I think we could have actually said airway, Control-Shift-M, assay, Control-Shift-M, head, and that would have done exactly the same thing. And we could even turn it into a tibble if that was something that we wanted to do. So anyway, so we've got this idea of transforming the data using this tidy syntax. So let's see, call data, airway as tibble. And then I wanted to use another dplyr and the tidyverse emphasize particular, I think they're called verbs, things that you can do with a tibble. And one of the things that you can do with a tibble is you can select particular columns. So I'm going to say, take the output of what I've got so far, and then select the column called sample, cell, and DEX. And so now I've got, instead of getting all of those columns, I've got a tibble with just the 3 columns that I selected. You notice that I didn't have to quote these things. I didn't have to say sample, cell, or DEX. I didn't have to quote them. So it's another kind of convenience that makes it easy to use the tidyverse. And then I just wanted to assign that to a new variable here, design, that may or may not use in the future. So yeah, so the tidyverse turns out to be pretty useful. I feel just a little mischievous here, and when you've done this, it seems very natural that since you're sort of reading the expression from left to right, it seems like the assignment should be at the end as well. Actually, that actually works. So it's possible to use this arrow for assignment, and it can be kind of fun to. It confuses your colleagues as well, so that's kind of fun to do as well. So you can have this flow of logic across, and then the last step in the logic is to assign the variable, the value that you've arrived at, to a new variable. All right, we better get moving on here. So I'm going to skip these next two little sections about another dplyr verb called count. And instead I wanted to look at the airway object again. We're really going to focus on these guys, the names of the rows, which are gene symbols. And I just wanted to extract the row data from the airway object and make them into a tibble using the row names as a column called gene. So this is the information about the genes in our airway object. The first one is ENSG blah, 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 3, 5, 419, 457, and so on. And there are a total of 64,000 genes in our airway data set. And these guys are the things that what we're interested in learning more about. So it's great to say, hey, I've got a gene ENSG003, but even an experienced bioinformatician will have no idea about what that gene does in biology. They have no notion of what that gene does. And so I wanted to figure out how we can be more informative about these genes. I'm going to take the next step here. Actually, I think now is a good time to just stop and make sure people are still on board, following along okay. Any questions?
Okay. Do you have any question? No response.
It's all right.
All good here.
Yes.
So far, so good.
Okay.
Okay,
all
great.
Right. So let me just take a quick look. I'm just concerned about all right, I guess we have to do this. So this will take just a couple of seconds, and it's a little bit tangential to the main point of things, but I actually wanted to do a DE-seq analysis, a differential gene expression analysis. So I'm going to start with this DE-seq2 package, and I'll say library DE-seq2. And for me, it loads. It might be that for one of you guys, it doesn't load because you haven't yet installed it. And so if you wanted to install it, what would you do?
Bioc Manager install.
Exactly. Install DE-Seq 2. And that would install the DE-Seq 2 package. Actually, I think the DE-Seq 2 package will have installed. If you've got the airway package, you'll have the DE-Seq 2 package as well, I think. So I'm not going to go into the gory details here. These are steps in an Rna-Seq differential expression analysis. So I'm going to copy these two lines, and I'm going to paste them into my R session. This first line sets the baseline level for the DEX treatment. And the second line says, I'm going to model the gene expression as a function of cell line and dexamethasone treatment. So it's like a two-factor experimental design. And I'm going to ask, with this line here, I'm going to perform a sophisticated statistical analysis that compares the levels of gene expression for cell line and dexamethasone treatment for each of the 64,000 genes. So we're sort of doing like an ANOVA, a fancy ANOVA, for each of the 64,000 genes with that line. And it's these comments that are being expressed, those are statements about each of the steps in the analysis. And the result of this analysis can be seen by this command here, which says, for this particular gene, ENSG, blah, 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 the average expression across all of those, all of the samples was 708 units. And that it was actually statistically significant differences between the treatment groups. It's not a multivariate ANOVA, ANCOVA. It's more like an ANOVA applied to each row independently. So it's like you did an analysis of variance or you fit a linear model to the first row, then you fit a linear model to the second row, and then you fit a linear model to the third row. So you fit 64,000 64,000 linear models. And probably you can understand that it's kind of weird. You've got 8 samples, but you're performing 64,000 statistical tests. And there has to be some clever approaches to make the best use of the data in that situation. It's actually really fascinating, and it's just a bit outside of the scope of what I wanted to talk about today. All right. Okay, so we've got the results, and I wanted to take the results and add them to the airway object. So I kind of wanted to update this airway object. So I'm going to take the row data of airway, which currently looks like no data, and I'm going to add to it the results of the differential expression analysis. I said oops, which means I ran into an error. I think this is my first major error of the presentation, so I'm doing pretty well, I guess. And it's also pretty easy to understand, at least as R errors go. It says object DDA not found. Here's the DDA, and I realized it was a typo. I meant to say DDS as the variable that I had created. So I'll just store that, and then when I ask about the row data on airway, I have information about the levels of gene expression for each of the genes, which is kind of interesting. So here's my airway object, and now you can see that I've added information to this object. So I know for each gene about its level of differential expression. This is kind of the end of the first chapter of the first real chapter of the book. And I wanted to save my object airway to disk. So I'm going to use this function save RDS, save my airway object, and I'm going to call the object airway with results dot RDA. And that just writes a single object to my disk. And if I look at dir, you can see, so that's like list files, the same command. It's just showing me the contents of my disk, and you can see that it's just created this object, airway with results dot RDA. It's just an object on the disk. All right. I'm going to take just a second here to switch over to the next chapter. What is an RDA object? Yeah. So, you know, it's actually a great question. So, we could have saved like this here, row data, DDS. We could have saved this as a text file, like a CSV file. So we could have said write CSV row data DDS, and then the name is like airway row data .csv. And then that's like a text-based file that we could have, you know, read in with read.csv, or we could have read into, say, Excel or something like that. But that's only like one component of this object, and I'd rather save a binary representation of the object so that you'll see actually in the next chapter. I'll just do this, rm airway. I've lost airway, and type airway, nothing there. But I can say read RDS and read in the airway with results.rda file. And it'll actually, I'll call it XX. And now I've got the object that I had before. So it saved it as a binary. RDA is a binary file format that's used to save just a single R object. So it saved just this single R object. It didn't save all of the objects in the workspace or anything like that. Like if I had chosen to quit, I think I can do this without, oops. That wasn't supposed to happen. I'll just be a second here. I thought it was going to prompt me to ask me whether I wanted to quit or not, but I guess I'd said that I said that I didn't want to do that. So let me just, that was bad. I was going to say that when you quit, you're asked whether you want to save. Yeah, after a while, it's got a will of its own. When you quit, it asks you if you want to save your session, and it saves it as a .rdata file, and that actually contains all of the objects in your current session. And usually that's not what you want to do. Usually you want to have more control over the objects that are saved. So I like using save RDA and then loading it in explicitly with load, or save RDS, load RDS, to save the binary version of the object and then load it in. So I think I'm back at presenting again with my RStudio. And I did that at exactly the right time, and it's exactly the reason why you save these things. I'd done a bunch of work, and I'd kind of finished it. I'd just finished chapter 2. I'd done a bunch of work, and I saved it to disk. and then I did something completely stupid and lost all of my work. But actually that's no problem because the central object that I need is still available. I can do airway is available as load RDS. Load read RDS, read RDS, airway with results.rda. And actually I'm back. You can see that actually when I load the binary object, it loads all of the packages that are needed to work with this object are loaded into my session, and I'm back where I started. So actually that was a wonderful
demonstration,
yeah, of best practices, and I'm glad I the best practices.
Best practices.
Yeah, exactly. So, okay, but that's that's great. So it's a lot of fun to do that. Right. So actually, I wanted to get into chapter 3 of this book. So again, this is at mtmorgan github.io/annotation resources. and I've just loaded in my airway data. And when I look at the row data of airway, I get the information that I had calculated in the previous chapter. And what I wanted to do was I wanted to make this just into a tidy format, and I wanted to take it, row data, airway, and I wanted to convert it into a tibble. And using this row named equals an RDA file is a binary file. It's just like the object in R. So if whatever the object was that you had in R, you saved it to disk. Like the summarized experiment object is actually really complicated. We just dumped it all onto disk, and then we used the only real use for an RDA file is to read it back into R with read RDS. So it's like this representation is not portable at all. You'd use a CSV file if you're going to share the data with someone who is using Excel, or you wanted to share it that way. Use Excel yourself or something like that. But if you just wanted, great, the binary representation for exactly what we did here. Yeah. Right. Oh, and then, right. So I wanted to turn this thing into a tibble, and it says I got my second error, could not find function pipe, and that's because we haven't loaded the dplyr package. And then, right. So yeah, as tibble. Tibbles are kind of interesting. They don't believe in row names, and they think the tidyverse thinks that a row name should just be a column in the data. And in the airway data, the row names are actually crucially important. And so we have to take the row names and include them as a column in the data, which I'll just call gene. So now I've got this. These are the gene identifiers that were the row names of the row data. They're now just a column in the tibble. And actually that's kind of good. So you don't have to worry about special treatment of rows. Everything's just a column as tibble. And what I wanted to do is, I think I'll get just a little bit more space here and see if we can't, right. Maybe even a little bit more than that. And just a little bit more, and then I'll probably have to make my, right, there we go. Right. So what I wanted to do was I wanted to know, well, which of my genes were most differentially expressed? And by way of most differentially expressed, I wanted them to have the smallest p-values, so the most significantly differentially expressed, which of these 64,000 genes were most significantly differentially expressed. And I'm actually going to use this column called p-adjust, P-A-D-J, an adjusted p-value, adjusted for the fact that we're making 64,000 comparisons. And what I wanted to do is I wanted to take this tibble and I wanted to arrange it such that the smallest p-value, p-adjusted value, shows up first, and the largest p-adjusted value shows up last. So I'm going to take my tibble, and I'm going to arrange it so that the gene with the smallest p-value is first, and then the next smallest, and the next smallest, and the next smallest. So these genes are these genes are most differentially expressed, at least in terms of the p-value. And actually, the p-value adjustment is just a Benjamini-Hochberg correction. It's actually straightforward to take different approaches to adjusting the p-value. It takes the p-value, adjusts for the fact that there are multiple correct comparisons, and reports the adjusted p-value. And I wanted to save this guy to a variable called top tibble. Sometimes they're called a top table, the most differentially expressed genes. So these are just top tibble, the most differentially expressed as a tibble. Yeah, and I'll just take top tibble and tibble. And I'll select the gene column and the p-adjusted column, and then we'll see. So whatever this gene is, ENSG, blah, 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 152583, somehow that's the most differentially expressed. And the biologists amongst us would be really interested in knowing more information about that gene. And so that's the goal. What more information can we find out about this gene? And what we're going to do is we're going to map or transform these identifiers into different representations. And in the bioconductor world, it involves finding what are called annotation resources. So these are packages that contain just data, but that map, for instance, between one type of gene identifier and another, or other types of information that help you to understand, place your results into a biological context. So I'm actually going to use this neat function in Bioc Manager called available. I know that I know a little bit about what I'm looking for. I couldn't quite remember. So what it does is it searches, like you remember that we were working with some package that looked like it started with something with air, but you weren't sure what it was called. So you could ask for, well, what packages in Bioconductor have, this is treated as a regular expression, have air in them? And you can see there's an air porter function and a package. I guess this is actually over Bioconductor and CRAN. So all of these packages, including the airway package, are have this air in them. So actually, it's pretty convenient. Bioc Manager available. What packages are there available that mention tidy? Not just in Bioconductor, but in CRAN as well. And there are all of these tidy packages. That's kind of neat. But actually, what I was looking for was a type of annotation resource that's based on annotations from Ensemble, which is the European center that provides annotation information, genome scale annotation information for the European research community. And it says that we have a number of packages that are based on the Ensemble database format. And the one that I want is actually this one here, ensemble.hsapiens.v75. And I want to use this annotation package because the airway experiment used an ensemble genome, version 75 of an ensemble genome, when it was doing a part of the Rna-Seq experiment. So I want to use this package. I'll say library ensemble. Again, you might not have it installed, and so what does one have to do to if you don't have it installed? You say Bioc Manager install ensemble DB. Better to cut and paste it. So that would install that package. These annotation packages have just a single object in them, pretty much. It's actually an object that's named after the package. So the package was called ensemble onsdb h sapiens v75. And here's the object. And it says, this object is actually a SQL database that was created at some time in 2017. It represents a particular ensemble version, and it annotates the 64,102 genes that ensemble knew about when this version of in this version of their database, and 215,647 transcripts. So it's a database that contains that information. Yeah, and if you get an error like Nera did, saying is not available, that's like, probably it was like, ensemble DB. So that's like, that's not that type of error, but it's probably, I guess it's like this type of error. Which says the package was not available. And that's a good question. So if you'd done install.packages and tried to install this package, or if you'd used the package manager in RStudio to install that package, then it would have failed. So Nira, I don't know, were you using Bioc Manager install to install the package? Yeah, then I'm a little bit mystified. The other thing would be, yeah, the other thing would be if there were mistakes, typos. It doesn't look like there are typos in what you've pasted, but if you had a stray space here, that's a possibility. I hope I didn't make another mistake here by doing that. Yeah, and then but then it actually, it says that. So I'm actually not sure, Nero, what the problem is. We'd have to troubleshoot it a little bit more closely. Yeah, so I bet, so I see that, I see that there's another, yeah, there's another error that Tena mentions about this, it's pretty cryptic, the message, could not create a temporary column for ... .1 is p-adjust. And I think that this is probably, I'm guessing here. So if we did row data airways, top table, try to arrange it by a column that didn't exist. It says, that's how I get your message. Yeah, so great, some questions here, yeah. Right, so, and the problem is that I guess your airway object that you used, Tana, wasn't the result of the first chapter. So it was like the row data was hadn't been updated to have the p adjust values.
We have another question. Why do you use row names equal to gene inside as table function?
Yeah, so let me do it this way. So this is just a slightly smaller example. I'm not sure that it's any better. So so this is the column data from Airway, and it's a data frame, and you can see that the data frame has these row names: SRR 1039508, SRR 1039509, and also, I mean, these row names are crucial for us, right? They describe the samples. They're kind of like the keys that relate the column data to the matrix of expression values. So notice those row names, and then when I make this into a lovely tibble, the row names have actually been dropped. So we've got sample name cell dex albut, sample name cell dex albut, but we don't have the row names, and this is because this is just the approach that the tidyverse takes to tibbles. They don't think that these data frame-like structures should have row names. It's like a philosophical position that probably makes some sense. It's like when you have row names, then you have to think especially about how do I work with row names as opposed to, well, I know how to work with a column, so I can just use all of my column knowledge to work with columns. And then you switch over to a row and you say, oh, now I have to use all my row names knowledge to work with row names. Maybe it would be better if you just had everything as a column. Then you'd only have to know call names knowledge. You wouldn't have to know row names knowledge. So that's the motivation for, I think, for the tidyverse saying, hey, I don't want to have any row names. And then the deal is, well, I've got an object that does have row names and I want to keep them. So you say, you add this argument, row names equals, and then you give it the name that you want. I guess here we'd call it like sample or something like that. And then what we've got are the row names have now become a column in this tibble that we can work with. So I hope that's helpful. Yeah. Also, in your post, Manuel, you said row dot names, but actually the argument is row names with no dot. I don't know why that is either. It's the tidyverse tries to be consistent, so elsewhere when it refers to row names, it'll definitely be row names with no dot. But I guess outside of the tidyverse, things are less consistent. Cool. So where are we at? Oh, on Stevie. I'm not sure. I think maybe Nera, we didn't ever solve your problem, and I'm not sure that I can really solve it in this forum, but I'm happy to help you afterwards. So we'll have to, I'm going to skip over this part. I'm going to skip, which is pretty interesting, but I'm going to try and get some actual work done. So we have this object, ensemble DB H sapiens. Oops. It's just typing in the wrong window. And kind of the central, a central idea of these identifier mappings is that you have one type of identifier, like these ensemble identifiers, and you want to map them to something that's more familiar or useful in some other context. And something like this ensemble DB package provides you with ways of doing that. So you can ask, well, if I have a particular, what types of identifiers can I use as the starting point for mapping to other types of identifiers? And so you say, you want to know what types of identifiers you can start with to do the mapping. And you can start with entree IDs or exon IDs or gene symbols or transcript names or protein IDs or this gene ID. And the gene ID is actually an ensemble ID. So you can start with an ensemble ID, and then you can go from an ensemble ID to any one of these other types of identifiers. So you could take, there's a function called map IDs, and you start with, you tell it where you want to find the mappings, and then what the identifiers are that you want to map. So the gene column, the ENSG, blah, 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 blah. What you want to map to, for instance, this gene symbol. These are the gene symbols. They might, if you're not a biologist, they might look pretty Greek, pretty foreign, but actually they're very familiar to like a biologist working in this field would, sort of have know by their gene symbol the genes that were relevant to the pathways that they were interested in studying. So what this does is it maps the ensemble gene identifiers to these more human readable gene symbols. And we'll look at just the first few of them by piping through head. So this first gene, the most differentially expressed gene in the top table, ENSG blah, 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 15282583, corresponds to a gene called SPARCLE, SPRCL1, SPARCLE1. And then the next most differentially expressed gene corresponds to the gene symbol CACNB2. So these, what we've used is these map IDs to map from the ensemble gene identifiers to their corresponding gene symbols. And we can put this into the kind of tidy framework. So here's our top tibble. And we actually want to map all of these genes, all 64,000 gene identifiers, to their gene symbols. So we take our top tibble and we pipe it to a function called mutate. So mutate is another tidyverse verb. And what it does is it changes the tibble. For instance, we can add a new column called symbol, and we can create the column by using map IDs with ensemble DB, using the gene column. We want symbols, and we're providing it with gene IDs. Now we've got over here, unfortunately, we'll need to, let me do one more thing here. Just, so, symbol. Symbol, blah, blah, and pipe it through select, dplyr select. I'll do this to get places and then come back and say gene and symbol and maybe p adjust. I've got mismatched parentheses here. So it's asking for a continuation. You continue entering stuff when I'd finished entering stuff. Right. So what I've done is I've taken my top table, I've mutated it, and then I've selected just the 3 columns. I've just selected them so that we could see indeed that this gene symbol, ensemble gene identifier, has been mapped to this gene symbol, human readable gene symbol, ensemble gene identifier, human gene symbol, and so on. And we've done this for all of the columns in the tibble. And so we now know that the gene with the most differentially expressed between treatment groups, which has this machine readable identifier, is actually the same as this more human readable gene, Sparkle1. And if we were biologists, we'd be totally enthusiastic to look at these genes and wonder whether they fit with the biology that we know about the phenomena that we're investigating, or whether there were some surprises in here that didn't make any sense. So we'd really like to explore that as a biologist. So I'm going to take, oh, right. There's one thing in here that I did that was kind of funky. I did this. I said dplyr : select. And does anyone know, first of all, what that does? What does dplyr : select do? Why didn't I just write select?
Because there is the same function in other packages.
Yes, so yeah, there may be select functions in other packages, exactly. And actually, so this says use the the select function from the dplyr package. But if you say get anywhere select, you can see that actually there are a whole bunch of different selects that are available. And Pinu says masked by DE-seq2, but actually it's masked by the select. In this particular case, it's masked by the select in annotation DBI, which defines select. And actually, look up annotation DBI select. Also, that's kind of interesting here, what I'm just doing now, maybe twofold. I'm using this question mark, which I find as a convenient way to look for help, as opposed to using this help browser in RStudio. Question mark annotation DBI select, and it takes me to the help page that documents what select does in the annotation DBI world. And I could also look up the help for dplyr select and find what dplyr select does in its world. And there are 2 versions of select. They're different. When we type select at the command line, we actually get the version that was loaded most recently. So because we first loaded dplyr and then loaded the ensemble DB, which loaded annotation DBI, the annotation DBI version of select wins, and it masks the select from dplyr. Yes, so that's the reason for using the : select. I'm just going to grab this stuff here, these lines, and paste it into my R session. And just walk through very, very briefly. I took my top table and I added a column for the symbols. And then I also added a column for entree IDs, which are another type of machine readable identifiers. And then in this select statement, I reordered the columns so that the gene symbol, the gene identifier was first, and then the symbol, then the entree identifier, and then everything else. So I was reorganizing the columns so that these identifiers were on the left of the table, and then the other columns were on the afterwards. And this everything is a kind of convenient way of saying, give me the columns, all of the remaining columns. You also saw that there are some warning messages. And it said, when I was trying to map from ensemble identifiers to entree identifiers, I actually wasn't able to map almost, or maybe even more than half of the identifiers. So there's like no entree ID that corresponds to the ensemble identifier, which is just a fact of life that different organizations who have developed annotations for genomes have pursued being successful or have adopted strategies that allow different numbers, different levels of success at identifying genes by their identifiers. So here's our top tibble. And we've now got these, we've mapped between the ensemble identifiers and these other identifiers. And I'm gonna do best practices and save this as a file so that I can pick up, when I accidentally close my R session, I can pick up where I left off in the next chapter. Are there questions?
I have a question. Can you hear me?
Yep.
Yes, this is Martha. So the question that I have is, what if your, I don't even know what to call it, the data set that you're working with is not already available in Bioconductor, like the species and things like that? Do you have to import it from somewhere?
Yeah, so that's a great question. And actually, it depends on how exotic your organism is. So Ensemble has, say, like 64 organisms, like the model organisms, and then organisms that are used pretty extensively in research. So like Caenorhabditis or Arabidopsis, or I don't know, rats, pig. Those they have this collection of organisms that that they annotate. And actually, if you're working with one of those 64 organisms, you're actually in luck. There's a package called Annotation Hub, a resource called Annotation Hub. And actually, we talked about this a little bit here, and I skipped over it, so maybe I shouldn't have skipped over it. And the Annotation Hub package has a single function called Annotation Hub. And what this does is it retrieves a database of annotation resources that we maintain on the internet. There are 50,000 different records in the Annotation Hub. And if you were to look for, query the hub for ensemble DB objects from release 101 of ensemble, for instance, maybe I should say, can use tricks here. You'd find that there were, that's too many. Let me try one more thing here. Yeah, you'd find that there were still too many, or maybe not. Maybe not, maybe I'm a little bit outdated. It says there are 243 Annotation Hub records for Ensemble DB, and then Ensemble releases every 4 or 6 or 8 months. So the most recent release is Ensemble 101, released earlier this month or maybe last month. And you can see that there are 243 records available. And maybe you're working on answer brachyrhynchus. Sounds like snow goose or something like that. It has this Annotation Hub identifier, and you could say Ensemble 101 answer, well, I'll call it Ensemble 101, is equal to hub double square bracket and then the Annotation Hub resource. And what it does is it runs out to the Annotation Hub and retrieves, just going to Google this, sorghum bicolor. Yeah, right. So I think you'll be in luck there. Footed goose. So what it's doing is running out to the Annotation Hub, and it's actually retrieving from the Annotation Hub the Ensemble DB resource, just like as though we'd loaded a package, but it's actually not loading a package. It's just retrieving the resource. And then we could look at Ensemble 101, and here's that object. And we could do everything that we've done with so far, we could do with this object Ensemble 101. So you work with sorghum, so let's try looking for Ensemble 101, and we're interested in sorghum. See what we've got here, see if we're in luck. I think we'll probably be in luck, but we're not. Yeah, so, okay, so now you're into the next level of difficulty, Martha, unfortunately. But not all is not lost. It's possible to make an Ensemble DB package and a
is that possible that for sorghum bicolor, the information, the annotation is in another package? Because I checked and there's a page for plant ensemble. So I don't know if it's in the same thing, in the same package.
It could be. So, it's great. So it could be that this function here, make Ensemble DB package, maybe it's actually possible using this function to create a sorghum bicolor Ensemble DB package. That'd be really interesting to look up. And I see Tana's pasted a link in the chat. So I'll just also mention that the place to ask about this, and you'll find really great and friendly help, and say, hey, is there an Ensemble DB package for sorghum bicolor? And you'll get an answer from the author of this package, Johannes Rayner. He'll probably answer, within a day, and he'll tell you the right answer, as opposed to me, I'll tell you the wrong answer. So yeah, it's also possible to build these things from scratch, as I'm trying to indicate. Make Ensemble DB package is one option. So I'll just paste that into the chat as well. Is one option that maybe you could explore as well. Do you know, we're coming to the end here, I guess, of the time, and I don't want to keep people too much longer. I'll say that there's still lots of material that we could cover. I think that you'll talk about genomic ranges with Stephanie next week.
Yeah, in 2 weeks.
2 weeks, yeah. I think I wanted to just end with one final thing that might be interesting in a kind of an advanced way, I guess. And I'll say this, here's this object, Ensemble DB, Reach Sapiens, blah, blah, blah. And it says that it has a back end, a SQLite back end. So there's like a SQL database that underlies this information. And I mean, it's a database you use when you have several things that look like data frames, but they're somehow related. You have so relational tables, and they're great for managing large data. So I just wanted to mention and explore just in the last few seconds databases. So when you have a database on the disk, you can create a connection to that database, and this dbcon function returns the connection to the database. It's stored somewhere on my disk in the package, in the installed Ensemble package. It has a SQLite extension. It's a SQL database, and this is a connection to it. And what I wanted to do was I wanted to use a package called dbplyr, which is dplyr but for databases. So if you know dplyr and the tidyverse, you'll actually find this to be pretty amazing. So you say src dbi, then dbcon, ons db, h sapiens, blah, blah, blah. And then assign this to a variable, tibbles without auto-completion. And it says, in this SQLite database, there are a number of tables: chromosome, ontrogene, exon, gene, metadata, protein. And I can extract from my tibbles one of those tables, like the gene table. And now I've got, in R, I've got something that looks, that's taking a peek into this database, and it's extracting one of the tables, or it's giving me a view into one of the tables where I've got a bunch of information: gene ID, gene name, gene biotype, blah, blah, blah. And then I can use all of my standard dplyr verbs to work on these things. So gene name in Sparkle 1, for instance. This says I've got competing filters functions defined as well. So here's the entry in that database for this, the Sparkle 1 gene. It's got a gene ID, it's protein coding. We know where it starts and ends. We know what chromosome it's on and what strand and so on. So actually, I'm not sure, we should probably make this a little bit more interesting as well if we just grab our top table here. this one here. So these are our two most interesting vector, the two most differentially expressed genes, and we've directly queried the ensemble database to retrieve information. And just here, I guess the kind of cool thing is that the first cool thing is that actually these annotation resources are implemented using a SQL database table. So that's kind of cool. And then I guess the next cool thing is that these SQL databases can actually be really useful for structuring large relational data. So that's the second cool thing. And then the third cool thing is that this dbplyr package gives us a very familiar way to interact with SQL databases. So if you do have some exposure to SQL databases, then using dbplyr can be a totally easy and refreshing way to access that data, especially if you kind of know a little bit of SQL, but you're stronger in R and the tidyverse, even just in R, then suddenly you'll find that it's really straightforward to work with these databases. So that's kind of like an advanced snippet that I just kind of wanted to share about databases. I actually feel like I've talked enough. I do have more material, but I think I've probably talked enough for the moment. And I'm happy to answer any questions or talk some more about specific topics that people might be interested in. And then to indicate that this resource here will persist indefinitely, and I'll actually try and add to it over the coming days and weeks.
Thank you, Professor Morgan. So please, if you have any question, please put your question in the chat. We have a question here. So, Janelle, wonderful talk. Do you have any general comments about the lemma package for differential expression?
Yeah, so lemma was developed originally for microarrays, and then it has functionality for working with Rna-Seq data. Actually, the package DE-Seq2 and EdgeR are really quite comparable in terms of is pretty interesting when they develop. They kind of leapfrogged one past another. One would have another new feature, the other a new feature, and now they're quite comparable. So lemma and EdgeR are quite comparable with one another. But a lot of the statistical complexity in EdgeR and DE-Seq2 focuses on the count data that's unique to Rna-Seq, that's different from like a microarray. But when you have a large number of samples, like maybe more than a couple of dozen, the unique features of count data aren't so important anymore. And you can transform the count data onto a continuous scale that makes the standard microarray analyses really appropriate. You gain very little by sticking in the DE-Seq world. So lemma has a function called voom, lemma. I'm not actually sure. Voom, I think. Yeah, voom.
I think it's voom, yeah.
Yeah, exactly. Yeah, so the lemma voom function takes the count data and transforms it onto a scale that you can use lemma on. And actually, if you have, as I say, it's appropriate if you have more than, say, a couple of dozen samples. But if you have like 60 or more samples, then DE-seq2 and EdgeR become painfully slow, and VOOM is blindingly fast. So if you have lots of samples, you gain very little by very little by sticking with DE-seq2, and you have the speed of lemma. So I'd opt for lemma and VOOM if you had more than a couple of dozen samples.
Thank you very much. So actually we have, could you send us all the R command lines, especially for debugger? It's very interesting.
Yeah, so I'm just saving my history now.
Yeah, actually the last tip was really interesting because I didn't know about it even, so about the SQL, how to, yeah.
So I've saved my history, but I don't know. Yeah, that's the dbplyr reference is great. Yeah, I don't know the best way to share my my file. I think what I'll do is I'll add a link to that page, to this resource. Probably in the prerequisites, I'll just add a little note here saying you can find the history of the session here.
Okay, thank you very much. So can we actually use the dbplyr for all SQL database?
Yes, actually, including SQL databases that are remote.
Ah, okay, that's interesting.
Yeah, so long as you can establish a connection to it.
Yeah. So thank you so much, Professor Morgan. Everyone is saying thank you. Thank you very much for this amazing workshop. I think we all learned so much today. So it was really a wonderful talk. I think we don't, do we have more questions? I think everyone is saying thank you. So
Yes.
thank you. There is no more questions.
Yes. Yeah, it's really great.
Yeah. Yeah, it was really amazing. All the,
Yes.
I think it was two, it's like Rna-Seq and Annotation Resources in one workshop. So it was, yeah, really wonderful talk.
Great.
So
We would like also to let you know about the third workshop of our R4Bia info series. It will be an introduction to genomic ranges and ply ranges. And so the workshop will be animated by Dr. Stephanie Hicks, who's an assistant professor in the Department of Biostatistics at Johns Hopkins Broomberg School of Public Health. She's also a faculty member of the Johns Hopkins Data Science Lab. So we'll be excited to see you later during our next workshop.
yeah.
Thank you.
So thank you very much for Professor Morgan for accepting our invitation and for this wonderful talk and workshop. Amazing, really amazing. And thank you for all the participants for attending and asking so many interesting questions. Thank you very much. We will keep you updated, so stay tuned, for our next workshops. And thank you very much. Thank you.
Thank you. It's great that you guys are organizing this. It's wonderful.
Actually, you accepted and you give us, it's like an amazing workshop. Really, we learned so much today. It's like 2 hours, but like, it's really wonderful.
Okay. See you all
Thank
later.
you. Bye-bye. Bye.
Bye bye. Ciao, ciao.
Ciao. Bye, everyone.